My name is Robert Kuiack, and today I'll be talking about my project evaluating the role of GraS and novel fatty acid metabolic pathways in promoting Staphylococcus aureus adaptation to antimicrobial free fatty acids of human skin. Staphylococcus aureus is a very interesting pathogen in that it asymptomatically colonizes 30% of the population. This is a serious health concern, as those who are colonized have a much higher chance of becoming infected once admitted to a hospital, and furthermore, it's usually the strains that they're colonized with that then subsequently infect them. So understanding how Staphylococcus aureus can persist on skin, even in the presence of acidic pH, unsaturated free fatty acids, and cationic antimicrobial peptides is paramount for new treatment options. Although there are a lot of pathways demonstrated here that provide resistance to Staphylococcus aureus, we became very interested in the sensor kinase GraS and what role it may play in responding to these harsh innate immune conditions. There are a variety of different methods we used in this project. The first being a growth curve, and this allows us to assess the growth of different strains of bacteria when confronting these innate immune conditions. As such, we created a variety of different knockout mutants, as well as mutants that had undergone site-directed mutagenesis to better understand the specific function of certain genes. Additionally, we used a luciferase assay to assess the expression of certain genes, and this is very effective as we can measure expression over the course of a growth curve. Finally, we used a cytochrome C binding assay to assess cell surface charge, and this is very relevant when studying GraS, as GraS results in a positive cell surface charge, and so using this cytochrome C binding assay, we can directly assess GraS activation. In Figure 1, what we see is a graphic representation of the GRA-XRS system. As you can see, cationic antimicrobial peptides or acidic pH are able to activate GRAS signaling. This leads to the phosphorylation of GRA-R, which then leads to the transcription of a variety of different resistance mechanisms. And together, these can create a positive cell surface charge, and this will repel cationic antimicrobial peptides. In Figure 1b, we see that there's an extracellular loop on GRAS that is absolutely essential for activation in response to cationic antimicrobial peptides. If we mutate these acidic residues, we get a GRAS protein that will be unable to activate GRA-R in response to cationic antimicrobial peptides. In Figure 1c, we see that although we can mutate this extracellular loop, seen as our GRAS 3D to G mutant, we are still able to activate GRAS signaling in response to acidic pH. And as you can see, this does not occur if we completely knock out the GRAS system. This is very interesting as it shows there's a novel sensing mechanism for our GRAS system. In Figure D and E, what we see here is that when bacteria is confronted with a cationic antimicrobial peptide polymyxin B, wild-type bacteria is able to grow just fine and our GRAS mutant is unable to. We can complement our GRAS mutant back with wild-type GRAS. However, if we try to complement back our GRAS mutant with this mutant form, the 3D to G mutant, we are unable to restore growth. This makes sense as the bacteria is unable to respond to the polymyxin B and provide resistance. However, in Figure E, while we see a very similar trend, we're now growing the bacteria at an acidic pH, which is able to activate our GRAS system, as seen in 1C. Now, even that mutant form of GRAS, that 3D to G mutant, is able to restore growth and allow the bacteria to grow in this inhibitory concentration of polymyxin B. While Figure 1 exclusively focused on cationic antimicrobial peptides and acidic pH, as I mentioned in the introduction, there's another innate immune condition found on human skin, unsaturated free fatty acids. And here we're using linoleic acid as a representative unsaturated free fatty acid. As you can see here, 50 micromolar linoleic acid is completely inhibitory to growth over an 8-hour time period. Anything above that obviously continues to inhibit growth. However, if we combine the other innate immune conditions found on human skin, acidic pH, seen in Figure 2b, or polymyxin B, seen in Figure 2c, we're able to grow the bacteria at very high concentrations of linoleic acid, 250 micromolar. This is interesting as the bacteria will face all of these conditions at the same time, and its ability to respond to acidic pH and cationic antimicrobial peptides and upregulate unsaturated free fatty acids pathways is a novel finding, and we're very excited about this. That we know acidic pH and cationic antimicrobial peptides can result in high resistance to unsaturated free fatty acids, we queried whether GraS plays a role in this heightened resistance. As seen in Figure 3a, knocking out conventional unsaturated free fatty acid resistance pathways, FARIR and FACA, had no real impairment of growth when the bacteria was grown under combined acidic pH and high concentrations of unsaturated free fatty acids. However, when we knock out GraS, there is a massive reduction in growth. Figure 3b takes this one step farther and again looks at our mutant form of GraS, our 3D to G mutant. As I showed in Figure 1, this mutant is still able to respond to acidic pH, and as such, we see this is sufficient to restore growth under combined acidic pH and inhibitory concentrations of linoleic acid, whereas our GraS deletion mutant does not fare the same fate. Figure 3c takes this one step further and looks at an effector protein directly activated through GraS signaling, MPRF. As we see here, MPRF appears to be required for this resistance. Interestingly, a single mutation, a K547A mutation, knocks out the synthase function of MPRF. For those who are familiar with MPRF, this protein produces lysophosphatidyl and then it flips this into the outer membrane. So we've knocked out the ability to synthesize lysophosphatidyl in this mutant, but the flip-base function alone, which remains in this mutant, is sufficient to provide this resistance. So this is interesting, as first it demonstrates MPRF plays a role in unsaturated free fatty acid resistance, and furthermore, it shows that the flip-base function of this protein might have been understudied, as it clearly has a role here. Now that we've shown Staphylococcus aureus can become incredibly resistant to toxic host-derived fatty acids due to this GraS system, we next queried whether the bacteria could actually use these fatty acids as an energy source. In a lot of other bacteria, such as Bacillus subtilis, beta oxidation is another way to get energy from fatty acids. While beta oxidation was not known to occur in Staph aureus, in Figure 4a and b, you can see the putative beta oxidation pathway in Staph aureus and its relationships to Bacillus subtilis, which has a well-documented beta oxidation pathway. Interesting, looking at the genetic layout of these operons, Staph aureus actually has all of its FAD genes clustered into one locus, whereas Bacillus subtilis has them on two very separate operons. Because this is a completely new area of investigation, we first wanted to get an understanding on how the FAD genes are regulated. When are they expressed? When are they turned off? What conditions may do this? In Figure 4c, what we see is that glucose really represses expression of our FAD genes. And this makes sense, as in other bacteria as well. Glucose is the preferred carbon source, so if glucose is present, it will repress expression of these beta oxidation pathways. In Figure 4d, what we see is that acidic pH leads to higher expression of our FAD genes, and furthermore, exogenous fatty acids, seen here as palmitic acid, can increase expression of our FAD locus in a concentration-dependent manner. Finally, we wanted to look at the genetic requirements for FAD expression. Seen in 4e, knocking out the FAD system, which we believe is responsible for beta oxidation, or FACA, another gene known to metabolize fatty acids, we see that there's an increase in FAD expression. And this makes sense, as likely there's a buildup of fatty acids in the cytoplasm, and the bacteria will want to metabolize these by expressing more of the FAD genes. Interestingly, knocking out PRSW or GraS results in an inability to respond to these exogenous fatty acids and upregulate the FAD system, indicating that PRSW and GraS likely both play a role in the expression of our FAD genes. In conclusion, we've greatly expanded our understanding on how Staphylococcus aureus responds to the innate immune conditions found on human skin. First, we've demonstrated a novel sensing mechanism for GraS, and this opens up the question, are there other conditions potentially encountered on human skin that may also activate this highly resistant pathway? Second, we demonstrated that activation of this GraS system, whether that be through cationic antimicrobial peptides or acidic pH, not only provide resistance to cationic antimicrobial peptides as was previously established, but also to unsaturated free fatty acids. Finally, we've demonstrated a bigger picture in that while the bacteria faces these host-derived fatty acids when colonizing human skin, there can be a lot of resistance that occurs through our GraS and our MPRF proteins. This then opens up the question, can the bacteria use these fatty acids as an energy source? And preliminary evidence seems to indicate that it indeed can, and we want to further investigate this more and assess any phenotypes that may come from this. Overall, Staphylococcus aureus is a very serious health burden, and understanding how it colonizes so much of the population is essential for targeting new treatment options that can hopefully reduce this serious health burden.